I'm delighted to introduce our first speaker, who's kind of going, he looks like he's going all TED Talk on me with his sort of, he's going to kind of walk around in a TED style. Hani Gabra is Professor of Medical Oncology and Deputy Head of the Division of Cancer at Imperial College. He trained in Glasgow and started his training in Edinburgh, which you won't notice when he starts speaking, obviously. He's also Associate Director and Head of the Cancer Division for North West London and an awful lot of other things, including President of UTROC. So I'm delighted that Hani has come. He's had a very busy weekend. I got an email from him yesterday because he wasn't sure where he was supposed to be, I think, because he's flying off to Berlin this afternoon to speak at the Charité in Berlin. So I'm delighted that you're here, Hani, and going to be looking at aspects of tumour resistance and heterogeneity, I think, is probably things you'll be touching on. So over to you, Hani.
Yeah, hello. I think actually it was Alzheimer's disease was the reason why I didn't know where I was and where I was going. And I guess that is the next big problem after we cure ovarian cancer with ultrasound. Yeah. I guess I'm slightly dissynchronous in this talk because actually I think originally I was going to talk tomorrow because I'm not going to talk about ultrasound. I'm a medical oncologist. And I guess that the reason that I'm here is for people to start thinking about how else you can use ultrasound, how you can integrate it into the wider management of malignancy, in particular ovarian cancer, which I'm going to be talking about today. And as I give this talk, which has absolutely nothing to do with ultrasound, I guess what you might want to be thinking about is how your skill set can map to the wider management of disease. And I'm not really going to talk about the front end, which has been so eloquently discussed by Dirk regarding screening and preoperative diagnosis. But I'm going to talk about the rear end of cancer, which is if we fail to cure it by early diagnosis, how do we manage this disease when it is in a phase where it is a chronically deteriorating situation. So just so that you know, here's a case history, typical case history of a 45-year-old woman who presents with abdominal distension. A CT scan shows disseminated peritoneal disease, and the best tumor marker we have, CA125, which isn't so good, was 2,500. The upper limit of normal being 35. And the most sure way of trying to decide what this is and deal with it at once is to go to a laparotomy. And as you can see, the disease that faces a failure to detect this early is a very disseminated peritoneal disease. How on earth total macroscopic clearance is achieved really is an amazing feat of excellent surgical skills. So extensive peritoneal disease was noted in this woman involving all her peritoneal surfaces. Partially cystic and solid ovarian masses were identified and removed. It's interesting that a typical presentation of ovarian cancer involves several kilograms of disease in the belly, but no evidence of disease elsewhere. A different situation from breast cancer, where very small primary tumors can be widely metastatic in the viscera. And she was found to have a typical histology, that of high-grade serous carcinoma, which affects about 60 to 70% of patients. And the immunohistochemical features were those of a typical cytokeratin profile, CK7 positive, CK20 negative, which is more of a Müllerian type profile. And again, a quasi-Müllerian marker, CA125, was positive and CEA negative by immunohistochemistry. So the typical management, which hasn't changed very much, involved debulking surgery followed by adjuvant chemotherapy consisting of carboplatin and paclitaxel, resulting in normalization of the CA125 and apparent complete response of the patient. And in those days, second-look laparoscopies were done, and this actually showed complete remission of all visible cancer. However, as so often happens, and probably happens in 80 to 85% of patients, the patient relapsed, in this case a year later, with gross ascites. The ascites was drained. She was retreated with the same drugs, achieved another complete response, relapsed, this time within 4 months, And this is the rub. She, at this point, was resistant to platinum, which means that the biology has changed, the probability of response for treatments is much lower, and she was retreated, in this case, at that time, with pegylated liposomal doxorubicin, progressed, and then died. And 25 years of treating this disease, this is more or less a very familiar pattern. So, in terms of how we manage this disease, we treat with primary surgery. We take the patient to apparent total macroscopic clearance, which of course in the vast majority of patients is not. There's microscopic residual disease. We then give chemotherapy to try and wipe that out. But in so many patients, the relapse occurs with a rising CA125 in an asymptomatic phase, followed by rounds of sequential relapse therapy, with chronic survivability ending essentially with the point of drug resistance. So we would define these, and I'm going to really focus on platinum resistance today, as a progression-free interval since the last platinum therapy of greater than a month and less than 6 months. So, however, that is a continuum, although it's a definition. And famously, George Blackledge, when he was working in Birmingham, he went on to lead a major pharma company, was asked by a pharma company, if we bring our drug and we give it, what response should we see in the patients? And his response to the pharma company was, what response rate would you like? And they were a bit baffled, but of course the answer is in this paper that he published, where patients who are relapsing within 6 months have a less than 10% response rate to platinum-based therapy, whereas those relapsing more than 18 months in his study had an over 94% probability of response to platinum retreatment. And so the time to clinical relapse predicts the probability of response. So in trying to understand this, sort of 2 million years ago, we created some cell lines in Edinburgh from patients with ovarian cancer who had ascites at the beginning of their disease process and then relapsed later with ascites again when it was platinum resistant. And we created cell lines in 3 patients where we have matched, isogenically matched, sensitive and clinically platinum resistant lines. Now the point about this is that 30 or 40 years of research into platinum resistance had adopted an approach where you take a sensitive cell line and you pour platinum on a Petri dish on it and infer that the mechanisms you generate in the plastic dish have any relevance to human platinum resistance. And indeed, 30 or 40 years of research have showed consistently that that was not the case. So these were actually very useful tools and have been adopted worldwide. Something that came out in a very interesting way was trying to understand how platinum resistance actually evolves. And as you can imagine, and these are not just aficionado points, you might give platinum and as a result, the cancer cell changes in some way to become resistant. So you have induced resistance in that cancer cell. And although this undoubtedly happens, much more frequently we are seeing, and certainly around the world there is an effort to characterize this now, we're seeing a very unusual and different explanation for platinum resistance, which is that the cancer cells are not changing. The cancer cells which are presenting are truly dying from the platinum, but what's happening is that very small numbers of clones are evolving and growing which are platinum resistant. And how do we know this? So from each of these 3 patients and from many patients subsequently, we find normal chromosomes in the presenting disease, which are then abnormal in the resistant disease, but much more importantly, we find very abnormal situations in the presenting disease and completely normal chromosomes in the relapsing disease. In other words, the relapsing disease could not have come from the presenting disease. These had to coexist and be selected for, whereas the main presenting disease is deleted. The recurrent disease, which is evolving and growing from a very small clone into the actual clinical recurrence, is a very different second disease. This has huge implications for how we treat platinum resistance, because it tells us that we may have to target are therapies against platinum resistance right at the beginning of therapy in the way that leukemia is treated in order to wipe out those clones before they ever evolve into a second disease. So we started to explore this process, and this involved taking these matched cell lines and undertaking microarray analysis and looking at the differences between each of the patients of the things that were upregulated by microarray difference analysis. And then, by comparing each of the differences, the deltas, across multiple patients, you get quite a small list of upregulated RNAs. And then we adopted a functional approach where we knocked down those RNAs, an siRNA knockdown functional approach, and we ended up with an exceptionally short list, which you can see here, which looks like 5 genes, but is in fact 3 clusters, which are folate receptor beta, HDAC4 STAT1 continuum, and the PI3 kinase AKT pathway. And I'm going to talk about the PI3 kinase AKT pathway essentially as an exemplar of how translational research takes you to the clinic. So some of you may not know, but AKT is a central molecule in the regulation of cancer. It has pleiotropic effects and exists in at least 3 forms, 3 isoforms in the cell, and is part of a pathway which is crucial in cancer and in life, which is an outside-in signaling pathway from the surface of the cell through the PI3 kinase pathway, the mTOR molecule, and AKT itself to signaling. As you can see here, PA1 and 2 are from one woman. PA1 is sensitive, PA2 is resistant cells, and you can see that we're using CDDP, which is platinum on the left, API2, which is an AKT inhibitor, and you can see in the bottom right, in the resistant cell line, peculiarly, the combination of platinum to which PEA2 is entirely resistant with an AKT inhibitor results in very obvious visual apoptosis. It's dramatic. The cells die much more so than sensitive cells, platinum-sensitive cells. So there's an addiction going on to AKT, at least in this woman, and indeed in all the women that we looked at in this system as an approach. And in the top right, you can see an isobologram, which is basically a mathematical approach that demonstrates the combination of 2 drugs in synergy. And you can see the data points here sit right below this line for synergy. In other words, there's fantastically strong synergy which occurs between the use of platinum and an AKT inhibitor in platinum-resistant disease. So that's an exciting but in vitro finding. So we utilize multiple distinct AKT inhibitors to actually show that this was not an off-target effect of that particular AKT inhibitor, that this was a common feature to all AKT inhibitors. And this is what we discovered, which was pretty crucial and actually has resulted in quite a lot of drug development, ending up now in several multinational randomized phase 2 studies in design. On the left, you see the cancer cells from a woman. The blue is the nuclei, and these are sensitive cells from that woman. And what we're doing at 0:30 and 30 minutes and 8 hours is exposing those cells to platinum. On the right is the same woman with platinum-resistant ovarian cancer, and you see that within 30 minutes of exposing the cells to platinum, there is this intense red fluorescence in the nucleus. And by 8 hours, it has leaked out of the nucleus as in the mitochondria of the cells. What is this? Well, actually, what's going on here is that in resistant but not sensitive ovarian cancer in the same woman, this molecule AKT is handled entirely differently. It rushes into the nucleus, which is exactly the wrong place for a cytoplasmic pathway to be. What the hell AKT was doing in the nucleus remains a mystery, but is very interesting. So there is a qualitative and clear difference between platinum-resistant and platinum-sensitive cells from within the same patient. In other words, a clear target to shoot for. So what we had inadvertently found from the patients was an entirely novel AKT pathway, where AKT is not handled on the left through the cytoplasm, but actually goes into the nucleus, is phosphorylated and activated as a result of DNA damage by an increasingly famous protein called DNA PK, which is responsible, it's a sort of yin-yang of BRCA. It's responsible for non-homologous end joining, but has an entirely separate life as a kinase, quite separate from its DNA repair function. So we did the obvious thing. We took a mouse. And I'll tell you a bit more about why mice are not so obvious anymore. But we took a mouse, because it's an in vivo system, and we engrafted on one side the platinum-sensitive ovarian cancer cells from that woman, on the right, the platinum-resistant cells. We then treated the mouse, and we used imaging. Finally, the word imaging emerges here. In this sense, in this approach, this is PET scanning that we did to directly image the impact of what we were doing in one mouse between two different cancer states, sensitive and resistant, from the same woman. And you can see on the right, in the platinum-resistant cell line, PO4, the line which shrinks is cisplatin with the AKT inhibitor. On the left, cisplatin and the AKT inhibitor shrinks the tumor, but cisplatin alone also shrinks the tumor. So, this is PET scanning, FDG and FLT PET, both done in the same mice. And you can see this crocodile jaw opening is the SUV inhibition of FDG and FLT PET, indeed, in platinum-sensitive disease. So this is what platinum-killing cancer cells looks like on a PET scanner. This is the resistant cell line. The crocodile's jaws are completely shut. There's no SUV inhibition over time, and this is absolutely resistant ovarian cancer. This is what happens to that position on the left when you add an AKT inhibitor and cisplatin. There is now clear evidence of quite dramatic SUV inhibition. In other words, we have restored platinum sensitivity. Of course, the word restored is a misnomer because these are quite separate resistant cells evolving slowly in the face of platinum treatment. They are intrinsically platinum resistant, not induced. So the conversation about restoration is a very inaccurate one in some ways. So we took this to GSK, and they bought it, amazingly. And we did a clinical trial where we started to give the GSK inhibitor, I think it might have a name now, but it had a very long number then, to patients with ovarian cancer. And this was the first translational study of the group called UTROC that we are now developing for translational research in ovarian cancer, and provides the signature platform for how we do those clinical trials. So, baseline CT scan, FDG PET. I said optional biopsy, but in fact, everybody got biopsied. And in our new series of studies, these are now mandatory biopsies. As mandatory sequential biopsies, pre-treatment and after 4 weeks on treatment, where we take 16-gauge core biopsies, fixed, frozen, and fresh. We gave various doses of, this is a phase 1 study, so we gave various doses of the GSK AKT inhibitor. And you can see here at baseline at 2 weeks and at 4 weeks, you can see a reduction in the FDG PET signal in various individual patients. This is not a biomarker for response. It has no relationship as to whether the cancer is shrinking or not. This is purely a pharmacodynamic marker because an AKT inhibitor blocks AKT and stops the sugar pumps working on the surface of the cancer cells. So this tells you that the drug is hitting the target, but tells you absolutely nothing about what happens to the cancer subsequently. So FDG PET is a pharmacodynamic, but not a response biomarker. And this, we knew this almost and confirmed it in the clinic, that we really would need to use FLT PET to get a sense of what's going on. Nevertheless, we saw quite reasonable GCIG responses, falls in CA125, just with a tablet once a day in these ultra-platinum-resistant patients, at least 3 lines of platinum-resistant therapy. And you can see here in blue these big drops in CA125 just with the tablet. This is not even with the platinum, okay? Just with the tablet, and actually some RECIST responses as well. But of more interest for you guys who are doing ultrasound, we managed to biopsy all the patients in this trial. We biopsied from nodes, subclavian, inguinal, para-aortic, from the liver, from abdominal and pelvic masses, peritoneal nodules, both ultrasound and CT guided, both at pre-treatment and on treatment at 4 weeks. And in 58 biopsies, we saw just one hematoma. Yes, she had a big bleed in her peritoneal cavity, but she was fine. Yeah, and we've gone on now to show that this is a very safe and acceptable procedure to patients. We then teamed up with Gordon Mills at the MD Anderson to undertake this amazing approach called reverse phase protein arrays, which I would recommend to any person wanting to capture information from biopsies. You can actually do this from 150 cells, almost from an FNA, but core biopsies did extremely well with this. And you can see here, where we drew a line for CA125 drop at 30%, we could differentiate the groups into what we called an AKT response and no AKT response. And the reverse phase protein arrays actually simultaneously looks at around 170 phosphoproteins. So there's a big snapshot of signaling centered around the PI3 kinase AKT pathway. And we could compare non-responders with responders and show that a whole bunch of pathways, as you can see on the right here, changed. A lot of groups of molecules changed as a result of the use of an AKT inhibitor. And we were able to cross-validate that. Here, for example, is the AKT. Here's in the bottom, non-responders. Before and on treatment, you see that the AKT signal goes from green to red. And you can see on the pathology specimen stained with phospho-AKT, this upregulation of phospho-AKT, which is a readout for a paralyzed AKT molecule that's been inhibited by the inhibitor. So we pulled out a bunch of response biomarkers, which either increased or decreased, and this allowed us to start to build predictive signatures for response to an AKT inhibitor. And maybe we can come back to that in questions. So we're now validating this signature, in fact, in a second paper, because what we did, and this is a trial that my colleague Sarah Blagden, who will be speaking later, undertook. This was the ACTRESS trial, which is AKT inhibition in platinum-resistant ovarian cancer. And this is when we put the animal experiment essentially into patients. We gave these patients exactly the wrong treatment, platinum to platinum-resistant patients. And we gave it with the AKT inhibitor. And we asked, what happens? So if I could just skip that. So this is what happens. This is a waterfall plot of CA125. And these are patients who are absolutely resistant to platinum. There should be no possibility that these people respond, perhaps a 10% probability. So we actually saw a 40% both resist response and CA125 response in these platinum-resistant patients, retreating them with platinum, which was quite surprising that we were actually able to see this in patients. So, and you can see here that patients are actually still on these medicines 250 days out from commencement. So, where do we take this? So we took this data to GSK just as it was being bought over by Novartis, and we found that this has become really prioritized now. So this has resulted in a 300 animal study to understand what the best combination of platinum with AKT inhibition is in order to design the next 2 clinical trials, which will be randomized phase 2 to phase 3 trials of both platinum-resistant and platinum-sensitive patients. And we look forward to that. Now, I guess the reason I'm here is that one of the things that we are unable to get a good handle on, as you saw from the FDG PET, is how to predict who's going to respond or not to platinum treatments. And whilst we have protein biomarkers, we've been starting to discuss and think about the approach of texturing, where we can couple the appearances of texture to biomarker approaches to try and better predict responders to particular drugs. And this could be something that actually we can develop more as as a collaborative approach. One final thing to mention is that we're moving away from the mouse model, and actually we're moving to this model, which is called the CAM model. This is the chorioallantoic model in the chick. So the chicks you can buy for £5. You can buy a dozen eggs, and you can put them like you would buy them in the supermarket in a little incubator. And I don't know if you know about Marmite soldiers, but you can cut the top off, and you can grow the tumor on the surface. And it used to be an angiogenesis model in the old days, but actually we're now using it as a xenograft model because these are living systems. And actually, if you get a robot arm, you can make it look like a BMW factory because you can have lots of eggs and a robot arm which is dropping all the tumor and then dropping all the combinations of therapy. And of course, if you buy a microscope or a PET scanner or an MRI scanner, you can start to image. And if you put a video camera on, you can actually do time-lapse and actually do video microscopy of what's going on here. So imaging has suddenly got very exciting, intravital in vivo imaging in a way that is highly scalable to look at drug development interacting with imaging is becoming a very interesting concept in our little hospital. So watch that space, and maybe next year we can tell you a little bit about imaging. Okay, I think that's, I know I'm overrunning a little bit, so probably, Tom, I'm going to stop there and take any questions.
Okay, Hani, that was great. Thank you very much. Now, there are 2 microphones. Who has the microphones? And we'll take any questions. Could the lights go up a little bit? I'm rather hesitant to touch the panel here before I switch off the entire AV system again. Thank you. Now, obviously, reached the age where I can no longer do such things. I have 2 people doing this. It's fantastic.
But at least you know which hotel you're going to.
Sorry?
But at least you know which hotel you're going to.
Yeah, exactly. Okay, I mean, obviously this is an imaging audience, so there's going to be an interest in some of the things you were saying about novel ways of looking at images and perhaps linking them to biomarkers to predict response. And I was just looking, and they are using CT histogram, texture analysis, and so on in other tumors now to try and predict response. And do you think it's sort of the, you know, you were talking about the pleiotropic effects of things, and how, do you think that's altering the phenotype of tumors, and that's altering the textures? I mean, what do you actually think? What do you think we're looking at when you look at these texture changes?
I'm not an imaging person, but I sit around with imaging people quite a lot, including yourself. And I guess I don't know the answer yet, but I guess we've been, for instance, we've molecularly profiled large numbers of patients with ovarian cancer. And it wasn't lost on us that we had actually pre-operatively CT scanned all those patients. And it's quite interesting that if you go back and you review those CT scans and you start to do formal texture analysis and try to cross-correlate that with outcome and with molecular profiles, multi-platform molecular profiling, such as RNA-seq, whole genome sequencing, reverse phase protein array proteomics, and metabolic profiling, you suddenly start to see, and I guess the false discovery rates will have to be fitted around that, but you start to see textures which are not discernible to the human eye, which can be picked up by these approaches that can then correlate either with response to treatment or prognosis of the patient. And then when you apply the omics, and I guess there is a term for it now, radiogenomics, something like that, you can actually start to see that particular pathways of genes can be correlated with particular textures on CT scan, which tell you about either prognosis or response to therapy. Now, if that is the case, and the radiologist can sit in the multidisciplinary team at the presentation of the patient and tell us how long the patient is going to live, what drugs they're going to respond to, simply from the texture of the CT scan, this could be outrageously important, you know? And I guess one of the other things that we would want to do is see if ultrasound would have similar aspects of texturing like this.
Yeah, I was good, because we've got Hongbo. You're here, aren't you, from the University of Buck? Do you have a view on this? I mean, because I know you've used SVMs, but I think you can use lasso as well. Is it lasso predictive models as well? So I wondered if you have anything you could sort of comment on in terms of the likely future of texture analysis with ultrasound, just because this is, I think, quite an interesting area. And whether you think these are, I think in C2 they've used different prediction and models. I mean, you've, as I said, SVM versus lasso. Can you comment on that?
Yeah, and it's a very interesting talk, first of all, Hani. And also we, in Buckingham, we have a lot of use of different kind of texture features. And Tom mentioned about the local binary patterns and in the spatial domain. We also have found there are a lot of text features in the so-called frequency domain. Sorry, don't want to go too technical at the moment. But this is an area, I think there are a lot of activities. And I would like to really talk to you sort of once after your talk, okay?
Okay, so the conclusion would be we think we might be able to use ultrasound in similar ways as with CT. I mean, on this thing of ultrasound, because I know that Antonia has done a lot of work on microbubbles as a contrast agent. And we looked at microbubbles, and whilst there are features of cancers that perhaps are different, diagnostically it hasn't added a lot with ovarian cancer. But do you see that maybe microbubbles and, whatever it's sonotropism, when you actually explode the bubbles in the tumor, might be a way of delivering drugs to ovarian cancer in the future? Because again, you can target the imaging and then perhaps target the drug at the same time. Do you think that has potential?
Yeah, I think it can have potential. It probably has the most potential for anatomically critical recurrences where if you don't get that bit of the tumor, the patient is going to die. For instance, something at the porta hepatis or something in the brain or stuff like that. Yeah.
Any other questions from the floor? I'm somewhat the one asking questions. Oh, here we go. Usha Menon, Professor Menon from UCL. We just get a microphone to her. Come on, Ruta. You can move faster than that. Come on. Let's see some. I know it was drinks at the PhD last night, but you can still move. Thank
you. Hani, whenever I hear you speak, I learn something new. But as far as the role of ultrasound, I think, one other area which is really important is where to biopsy.
Which biopsy?
Where to biopsy. When you're taking the true cut,
yes,
at any point, which part of the tumor because of tumor heterogeneity.
Yes.
And if by ultrasound you could designate two very different areas so that you might get the dominant clones as well as the majority clones up front, because sometimes you will not get the dominant clones up front on that biopsy. And I think that is where one of the areas, so instead of, in addition to prediction, really this directing the biopsy on texture, yeah, or some kind of would be very helpful.
Yeah, so I'm a fantastically Darwinian individual, and I guess Imperial makes you that way. I do think that, cancer presenting in an individual is a whole ecosystem. And, it's as representative as all the people on the planet and all of speciation on the planet. It's a metaphor. So if you apply those principles, you're hunting for rare events right from the outset. And so it strikes me that no one technological approach is going to, safely cover all the bases of such a complex ecosystem. And we're probably going to have to take a multiple biopsy approach, as well as differentomics, as well as an approach to depth of sequencing and depth of analysis, which will give us an overall view. Maybe at some point we're going to have an algorithm, an omics algorithm for cancer from the outset, which will tell us about the probability of these rogue cells causing problems in the future and a probability of that happening. We call that prognosis at the moment, but I think that will be much more refined as time goes on. Yeah.
Hani, thank you so much. I think we're going to have to finish. I think that we could carry on talking about this for a long time, but I think I can see that the sort of medical oncology and imaging is perhaps coming together. Could find some interesting developments in the coming 2 or 3 years, I think. I hope you're going to be around at least for a few hours before you fly off to Berlin, so people can have a chance to ask you questions over coffee.